So that was the story of how I got started with research. And it couldn't have been any more exciting than that. Fast forward a number of decades, and I've been, as you heard, at this place called Sanford Burnham Crevice. And I have used, as you might guess, PCR an awful lot in my lab because of the new things that I've been working on. I want to call them new for about the last 20 years. And these were human disorders in the process of glycosylation, like adding sugar chains onto protein. And so before I jump into the sciencey part, I sort of want to give homage to somebody who's very important here. And how do I get this thing to advance? What do I do with it?
Click on these little arrows here.
Oh, that one, I got to go up to that. Okay. All right. So this is Dionysus, the god of the harvest. And I was lucky enough to see the reincarnation of Dionysus in 1981 in the person of Dr. Addy. So we were at a grape harvest, and you can see him very, very much enjoying the grapes that were coming along. So he was accompanied by his attendant, who then followed suit and was able to drink some of the same stuff. And that person went on to become an extremely successful biotech entrepreneur, I guess you would say. And he was a guy that we both knew pretty well. So Alan and I go back a long way, and I hadn't seen him for many years. And just again this summer got to reconnect, and he can tell you if he feels like it about the Stanley Kowalski thing. He actually started off, instead of greeting me, he used a line out of Streetcar Named Desire to say, "Are you familiar with the Napoleonic Code?" Anyway, read the play, you'll understand. But let's get on to the other stuff. You know, this is the way we think it works in science, right? You start out with basic laboratory research, there's some translation that goes into clinical research, and in the long run, it improves health. Well, this is something Larry Tabak said. He says, you know, but it really doesn't work that way. And Larry Tabak, as you know, is the interim director of the NIH. It's a circle. Each side influences the other. Each side contributes. We are in a matrix that we are privileged to be able to be a component of as working in some aspects of basic science, and hope that we make some differences in people's lives. And so I study these things called congenital disorders of glycosylation, CDG. And I say there is an ecosystem here, and that is the physicians and the basic scientists collaborate, probably not enough. We all don't mix it up with docs as often, perhaps, as we should, but there's a really important component in this, and that important component is the families. And because usually the interaction is physicians and families and patients, but because glycobiology was not and is still not well known by basic scientists and physicians, I happen to be fortunate enough to be in the position of being able to act as a bridge between them. And so many of the families would actually come to me and a couple other people who were involved in glycosylation and say, can you help me with our kids? We talk to our doctor, the doctor doesn't know anything about what we're talking about. And I felt that at that point, we as basic scientists need to be able to give something back to the families. And so we completed the ecosystem in all of the contacts between everybody. And you know, if you don't know much about glycobiology, don't worry about it. That was from a movie called Extraordinary Measures, Harrison Ford. He played actually a guy I knew and worked a little bit with in the lab with Stuart Kornfeld. So we started off this venture by running into this phone call I got from a doc. Boy is probably going to die. Can you help us save it? Wow. I'm a basic scientist working in the lab, and a doc calls me and says this. You can't quite avoid that, but you don't need any more drama in your life after you hear something like that. So we'll come back to that in a few minutes, and we'll follow up how that worked. So, this is something that folks may have seen before, but perhaps not. These are an assembly of the different kinds of glycosylation pathways that exist in mammalian cells. And I think that here we have, rather than boats and chairs and alphas and betas, we make the sugars just these little symbols. And it is easier to track. And so you can see there are different pathways that contribute to proteoglycans like heparan sulfate, chondroitin sulfate, glycoproteins that are the kinds that you might be familiar with here, because all secreted proteins, or almost all, and all membrane and matrix proteins are all N-glycosylated because they go through the endoplasmic reticulum and then onto the Golgi for their secretion. There are also other glycans called O-glycans, such as those here. There are still other ones that are linked to the proteins in different ways. It can be a linkage through fucose. It can be a linkage through glucose. It can be another kind of linkage through fucose. So there are many, many different possibilities. There are probably several hundred enzymes, proteins that are important in glycosylation. But in doing that, there's actually, come on. Why don't you listen to? There we go. So they have these things in common. If you're going to use sugars, you've got to activate them. And those sugars can come de novo from other sugars, or they can come from salvage of turnover of proteins and sugar chains and lysosomes, or you can make them from something that you get from the diet. But in any event, it requires that there's a glycosyltransferase, in other words, a transferase that will take an activated sugar donor, nucleotide sugar, and transfer that using the transferase onto a protein. And that is usually done inside the Golgi and sometimes in the ER. And all of those things, this is the critical part, have to be co-localized to have the reaction go. So the sugar transferase, the donors and the acceptors get one time. And that's the product. You need metal ions, you need the right kind of vesicular pH to have this to work. And so if you look at the discovery of different congenital disorders of glycosylation, now these are sorted by different pathways, and the biggest one that I'm going to talk about here is the N-glycosylation pathway, which is this, and this is the one, as I said, uses the ER and the Golgi. And you can see that well over half of the disorders, now approximately 90 of them or thereabouts, are affected in the N-glycosylation pathway. So this, if you think about it, even if you don't know about glycosylation, you can see here that there's almost 170 different genetic disorders that affect the addition of the proper sugar chains to proteins, their modification, their buildup. And so starting with that, we have more detail that we could go into. We're going to keep it very short because I understand that people may be not, oh, by the way, if you're going to treat patients, you're going to have to know the natural history of that disorder, some biomarkers before you can get to a potential therapy. This is a diagram of a schematic of the N-linked biosynthetic pathway. What you need to know is that every place there is a red indicator here. That's another gene that has been mutated in the assembly of this sugar chain. It starts off on a lipid called dolicol, and then one by one, the individual sugars are added. And as you can see, there are a lot of green things that are being added, and those green things are mannose. That mannose, once it's matured, is transferred onto protein, and then of all things, it begins to get trimmed down again. It starts off with a lot of mannose, and then it loses mannose progressively, and then there is the addition of other sugars onto that glycan. Now, this is a generic representation. And in fact, if you had all of the structures, there would be hundreds on here. So this is a generic version, but it gives you an idea. So what can we do with this? Up here, we're going to look at not most of this part of the pathway, but just the area up here that involves the sugar precursors. So what will that do for us? So here was the little boy. Can you help us save him? So this patient had coagulopathy, so he would bleed, but he was in danger of clotting. His coagulation factors were all off. He had hepatic fibrosis, so there was fibrosis going on in his liver. His liver enzymes were elevated. He wasn't doing well. He was a very, very small child, 6 years old, but just not growing. And he had this thing called protein-losing enteropathy, which I had never heard of. I mean, I'm no gastroenterologist. I don't know what this is, but it's proteins leaking out from the circulation into the gut. And they do this because the seals between each of the individual cells in the intestine are not solid. So it's paracellular leakage. And so the reason this guy called me was because he had seen a paper that we had done. And the paper said this: we have this glycosylation disorder. We don't know what the cause is, but we could just take the sugar mannose and we could add it to these cells that didn't glycosylate things well. And now if you just added mannose, it would improve. Well, wouldn't it be great if you could just do that for patients? How about you just feed them some mannose? And I decided to go to the FDA and say, there are no human trials out there, but it's just sugar. Come on. Will you give us permission to do it in my lab? So the phase 1 trials of mannose feeding people is our lab. So we lined up everybody over 2 weekends, and we had various doses of mannose. We tapped our blood. We looked at the kinetics, and we had that study done, completed. 2 days later, that's when he called and he said, do you have any idea how much mannose to give? Because I think we're going to lose this kid. And I said, yeah, give him this much, this often. And here's what happened. Before, this is AT3, it's a coagulation factor. Here's the normal level. Before mannose, it's down. Afterwards, it's up. Albumin, which he had to have infused into his blood to keep it up high enough, here he is always low. After this, he's normal. Fecal alpha-1 antitrypsin here is high, and that should be low because that's an indicator of leakage from the gut. And so what we see is after going on mannose, he comes down and he's normal. Wow. So I went there to tell the doc, look, this is in Munich. You got to take this kid off mannose so we can see if it comes back. And you can imagine what the doc said to me. I said, I am not disturbing this patient. This is as healthy as this kid has ever been in his life. I'm not taking him off. So then we go down to the beer garden and we're thinking, what is it? Is there anything we can make sense of? So I had the fibroblast in this kid in the lab. And I remembered one experiment. And the experiment began to tell me what was wrong. And you know what? This guy knew it. What was that? He said, mannose 6-phosphate. I couldn't believe that we got him to say these things. It's true. So what's the deal with mannose 6-phosphate? Well, here is mannose 6-phosphate right there. It's on the way to making glycoproteins. So how is it that mannose could help this reaction? What was going on? Well, what we saw was, in fact, that the fructose 6-phosphate to mannose 6-phosphate, we suspected that could be at least one of the possibilities. And then we were able to figure it out because we used 2 tritiated mannose, radioactive mannose, to label the glycoproteins. But what's important about this is if mannose comes into the cell, is phosphorylated by hexokinase, and then leads to glycosylation, it's in glycoproteins. But if it goes to glycolysis, the tritium splits off as water. That water then equilibrates with outside and is never coming back. And so what we noticed is that the amount of water that went into essentially glycolysis was down. It was far below normal. And I said, the only way we can explain that is a deficiency of phospho-mannose isomerase. And that turned out to be the defect. We were able to then do the enzymatic assay and to figure out what was going on. So this is a little schematic of the pathway. And what you can see is that this, please go that's where the lesion is, right there. Okay. That's where we had the lesion. The therapy then was just mannose would go in, go around the block and completely restore the normal synthesis of the lipid-linked oligosaccharide. Even though about 95% of the mannose that goes in is actually burned in glycolysis, elevating the plasma level from a normal level of about 40 or 50 micromolar up to 200 micromolar was enough to rescue this kid and to make most of his glycosylation normal, as you saw. Only a couple of percent goes into the glycoproteins. So it's a matter of where you are in the pathway. And, you know, he had many different problems, but almost all of them were corrected by mannose. So if we're going to do the right thing as scientists, what are we going to do? Well, we're going to make a knockout mouse, right? All right, make a knockout mouse. And you know what happened? The mouse died. We got no viable offspring. What should we do? Students, what should we do? Can we do anything else? What did we do for Max? What did we do for the patient? Give him mannose. Yeah, give him mannose. Sure. But, you know, we figured we'd give it to mom and then mom would go ahead and give it to the kids. And so there we did. And guess what happened? The mice died earlier. We couldn't even find any remnants. So what was going on? Well, it turns out that if you completely make a null, you accumulate mannose 6-phosphate because it has no other place it can go. And when you do that, you deplete ATP and now you're in trouble. So then, as you were saying, why don't you just make a hypomorphic mouse? Well, we did that and it didn't have all that much of a phenotype. It had a little bit of one, but when we knocked in the patient mutations, here's what we saw if we gave it during gestation. Look at the mouse on the right. The mouse was born without eyes. And what happened? And then after birth, if you gave mannose, you had this cloudy eyes coming up like cataracts. Well, this one, the cataracts, we could understand because too much mannose can lead you to mannitol, which is similar to galactitol, which is the problem that you get when you have galactosemia patients. Okay, so there is, and why in the eyes? Because the amount of enzyme in the eye is the lowest of anywhere in the body. And it's so low that under normal conditions, it can handle it. But if you put a load of mannose onto it, now you've exacerbated the situation. And so now you're in trouble. And even though this is only 2% in the drinking water, it really made it ugly. So what was it? We could see that this is normal eye development, where we won't spend much time here. This is at 10 and a half, 12 and a half, up to 16 and a half. And you see that there is the development of the lens at that point. Well, turns out that in the knock-ins that had about 8 to 15% activity, the lens never develops. And you have this empty space that now causes essentially the eye to collapse. So that's our story on mannose. And it didn't stop us there, because we are interested also in other sugars. And now I'm going to get into something that 3 people in my lab just did a wonderful job on. And I'm calling this metabolic heritage mapping. And that's where the idea of doing biochemical archaeology comes in. So next kid we ran into was Abdullah here. So he had hypotonia, intellectual disability. He was a very, very small kid, short arms and legs, and he had elevated white cells. His blood looked like Campbell's tomato soup made with cream. I mean, it's just creamy looking stuff. So what is wrong with him? Well, we could see that there was actually a deficiency in the addition of fucose to something. We could see that was absent. So we thought maybe what we could do is the same thing we did before when we were drinking mannose. Let's try at drinking some fucose. Along with Thorsten Marquardt, we did the first ever human trials of fucose. He drank some, I drank some. We did this little titration, and this is what we found. So what do you think happened? Did we succeed? Look at this. Here are his neutrophil counts. The gray area at the bottom is the normal level. And you can see that up in here, he's way, way above. And within 3 days of starting mannose, he's back to normal. Now, his intellectual disability and growth and all didn't correct, but this one did. So what was going on? How did this so effectively happen? Well, turns out that he lacked a molecule called silyl Lewis X on the neutrophils. And that allows neutrophils to roll on the surface of the endothelium and then to extravasate in areas of infection. He couldn't do that. We found out eventually that his deficiency was in a transporter, the GDP fucose transporter that's located here, and that takes the GDP fucose, which is made in cytoplasm, into the Golgi. And there is a de novo pathway to get to GDP fucose. There's also the salvage pathway. And what we realized is that this actually, believe it or not, should not have worked. This was the information from 1975 that said 90% of all the fucose comes from this de novo pathway. Only 10% comes from exogenous fucose. So how did a few grams of fucose distributed over this boy's body everywhere make it to enough to go to the bone marrow, to hit those cells, to give them silyl Lewis X, and to now suddenly have enough that his neutrophil counts fell? That should not have worked. So we decided we're going to go take another look at this. And we began to think about the big metabolic chart that you've all seen before, this big gamish of everything. Well, that's, if you're going to do that, that's precursors, right? That's all the reactions. And it's this nice flat, put it on your desktop and take a look at it. But I maintain that the metabolic chart is not flat. It's got peaks and valleys, and it's got a lot more detail. So if you're going to make glycoproteins, you can take sugars in from the diet, you could take and make them de novo, or you could use it from salvage. And what we had seen, or what the assumption was that all of these things are together in a big mixture, that are just floating around in this, literally this pool of cytoplasmic activated sugar nucleotides, sort of like this, sort of like a collection of Lucky Charms. That leads on to glycoproteins. But we began to wonder, this bedrock biochemistry only used one cell line. They did it at one fucose concentration. Maybe that was not altogether the right one. They were doing metabolic tracers, but what if the metabolic tracer is not really only a tracer, but and it's not revealing everything, but it's really the substrate, and the substrate being handled differently. So we thought about this, and that 90% de novo and 10% from there. Nobody ever thought about what's going on in the lysosome. How could you measure any fucose that's coming out of the lysosome? So we decided to really go into there and show that cells can selectively use fucose from different sources. So again, here's the 90% de novo pathway that comes from GDP mannose to GDP fucose versus the diet and salvage pathway, it's 10%. So when we did this, we labeled HepG2 cells, and we did this for 10 other cell lines. We looked at supplying exogenous fucose. Now we increased the amount and went up to 50 micromolar. And as you can see, that which is coming from fucose becomes dominant, and it suppresses the amount that's coming from both glucose and mannose. This was something that, we had not quite expected, but it confirmed that the method before was just not complete. It was well done, but not complete. So if you look at it and, sort of spread out like this, you can see that this is what it will have the appearance of. We noticed something else though, because we knew that glucose and mannose, making GDP mannose, would then go into glycoproteins in mannose and stay at that same relative proportion. We always saw that. So we thought, coming from here, that there would be GDP fucose, and regardless of the amount of exogenous fucose that would be provided, you would still get the same amount going in. And we were surprised to find out that that was not the case. If they're all in one pool, they're all going to be handled identically. But what we found was, if you list the 100% of each of those, you can see that glucose goes down much faster than mannose. So again, glucose to GDP mannose to GDP fucose is different than fucose that has come from mannose to GDP mannose to GDP fucose. They're handled differently. And we think that that could mean that there would be separate pools of these guys. And looking at it that way, this is what we would have predicted, but that's what we determined. So we thought that there were probably different pools. Now, what was interesting is then the next step for us was to take cells, pre-label them with heavy isotope of fucose until we saturated all the sites. It took 10 days. So now all the fucose that's coming from the cell that could be salvaged would have a heavy label. We used a different label to provide fucose that would come from the outside. We used a different label for mannose. We used a different label for glucose. So now we can measure the simultaneous contribution of all these different pathways all at the same time. And we're able to do it because we looked at secreted glycoproteins. And we're able to isolate the fucose from that. But by doing that, you have one chance. Donor, acceptor, transferase. It's a one-way street. And then it goes outside. And what we saw was that we could distinguish all these different pools. And if you look at it this way, you can see again that glucose and mannose are handled differently. But look at salvage protein. So salvage has to use the same enzymes to be activated and donated as fucose coming in from the outside. Somehow the cell is saying, I want this fucose, not that fucose. I want to get rid of that fucose. So this suggested to us that there were really some kind of segregation of pools that were important and that the cell knew about. And we tried this in different cell types, but we didn't know the basis. And I have to confess, even at this point, we don't know it. We then went on using a different cell line to look at how lectins could be used to score the amount of fucosylation. So lectins are able to recognize the incorporation of fucose into different linkages. For instance, LCA recognizes the fucose down here. UEA recognizes fucose in that kind of linkage. And in this one, in LTL. So again, different kinds of modification are being using fucose from outside in different ways. Now, these are just within the cell. This is not looking at secreted stuff, but we could see that there was a preferential use of fucose that went to different kinds of glycans. Then we did another experiment where we looked at those molecules that were labeled within the cell, either with C12 fucose from any source or C13 fucose from outside the cell. And we did this in 6 cell lines. And we found that because we used M+6 fucose from the outside, that this saturated, and we looked at hundreds, as you can see here, hundreds of glycans, that this saturated at about almost 20 micromolar. Now, if we looked at, because we could LCMS separate all the different glycans, we could see that each one had to have either C12 or C13. You know, one shot, that's all you get. Now look at what happens trying to get labeling 2 fucoses. There's a lag. So again, the first fucose, which tends to be this one that's here in the core versus these other 2, has a preferential use of exogenous fucose. So again, you know, pictorially, it looks like this. We did this also by doing mass spec analysis, fragmenting the glycan, knowing where exactly the things came in. And so we could show that, in fact, it was here and it was behaving the same way by doing it by mass spec, not just the way we were doing it with lectins. So again, it's this. Then we said, well, look, what if we could change the location of the cell where fucosylation is taking place? So there are 2 kinds of fucosylation, again, the red triangle, that take place in the ER. One is specific for Notch proteins with a certain sequence homology, and that's catalyzed by POFUT1 protein oligosome fucosyltransferase. There's another one that is recognized in Adams TS is TSR repeats, and they are doing the same reaction, but on a different set of proteins. So we provided the artificial substrates, we looked at the secretion. And again, we could measure all 4 different contributions to its secretion. And here's what we saw. So I think you've seen this kind of picture before, but let me put it in this way because I think it works easier. Now, if you compare POFUT1 and POFUT2, you can see that, for instance, POFUT2 prefers to keep the level of turnover proteins. Again, these were pre-labeled. So salvage stays up high. POFUT1 tends to go down. And fucose, of course, goes up in all the cases. But you notice here, again, now glucose and mannose overlap. They're close to overlapping here. And here, the N-glycans, all the other glycoproteins that were made, they still stay separate. So we have a situation where again, cells are deciding on different choices for adding fucose onto different proteins made in different places within the ER. How they do that, we don't know. So this is our sort of general schematic. Here's the activated sugars as the big slushy pool. But based on the data I've presented, maybe they're more like this. Maybe they're segregated pools of Lucky Charms. And if you go back to the big metabolic chart we were talking about before, maybe you excavate and look more closely. Maybe it's like this. Maybe this is more like, this is the DC Metro. So there are ways of getting in the cell. There are places you go to exchange and go a different direction, but you don't have free entry at any point you want to. And I think that's the general thought we have about the cytoplasmic pools of things. They're sloshing around. I don't believe they are. I think they're segregated, they're separate. We don't know the basis for the separation, but we can see that there must be ways that the cell is telling us, yes, even though I'm going to use the same enzymes in this reaction, I don't let them mix. There is diversity, there is not equity, and everybody is included. So we began to wonder, how does fucose even get in the cell? And so we designed an assay, a lectin binding assay, where you have no color, and as you add fucose, you can see that there is color. All right, that's great. We do this in 384 well plates, and we knock down all kinds of different transporters, about 160 different transporters, so we could see what is preventing fucose from coming in the cell. And what we found, look at the blue arrow on the bottom, was that in fact, it was SLC2A1. So, okay, what's that? That is the major glucose transporter, GLUT1. Wait a minute, it's a glucose transporter. What the hell is it doing transporting fucose? We don't know, but what we did find out was that there is no competition with glucose. We would put in 5 micromolar fucose, 25 millimolar glucose, said, I don't care, I'm going in the cell. So fucose was totally insensitive to that. We tried a few different cell lines. So again, somehow the cell is recognizing that it can bring fucose in. And we also noticed that there is another way that fucose can come in, and that is by macropinocytosis. And if you look over on the left here, we had different inhibitors. This is the control, and what is that, 876 is a highly specific GLUT1 inhibitor. And you can see that at low concentrations of fucose, 5 and 10 micromolar, you can inhibit about 50%, which exactly matches what we can do if we use GLUT1 knockout cells. Here's the Western blot, and you can see this is completely gone. So this fits with what we see at low fucose concentration. If we use amiloride, which is an inhibitor of macropinocytosis, we knock down independently of GLUT1, we knock down a lot. And then if you combine both together, you have, why doesn't that show up? Is that showing up there? I feel like the blind man here walking around. All right, there's the two together. But notice what happens as you go up in concentration of exogenous fucose, that suddenly that is no longer significant. So remember, this is being added to cells at higher concentration, and it's coming in by a different pathway. So we believe that that's at least one of the ways that we can begin to differentiate fucose coming in from the outside versus fucose being reutilized. We're working on that. So we began to think about what are the possibilities of doing proximity ligation analysis of the enzymes that are involved in the salvage or exogenous pathway. Let's do a proximity ligation analysis of those enzymes and see if they're together. And what you can see is without fucose, they're scattered all over the cells. And then if you add 50 micromolar fucose, now you reduce the number of those aggregates or those interacting molecules, and they tend to change their localization. We're at the very beginning of this result of a month ago, no, 2 weeks ago. So we don't know what this means, but it says that those enzymes are aware of each other and they respond to exogenous fucose. Might they respond because there is in some way a control between changing the levels of fucose inside the cell? And you say, I'll bet all this can be explained by just increasing the GDP mannose pools. That's what you're doing. The pools stay constant. The cell is absolutely committed to keeping those pools constant. So we wonder if some of this, and perhaps even those activating siRNA knockdowns, really have to do with whether a fucose comes in, from which source, we don't know, and it goes to degradation versus one of the pathways. Is the degradation specific for one origin? Because remember, that coming out of the turnover pathway, the salvage pathway, that doesn't get hit. That's protected. So again, there's some way the cell is knowing that they've got something that is unusual. But wait, there's more. GDP fucose, we've talked about, but here is our old friend GDP mannose, and what you remember is that can come from two different sources, but there's also a thing called dolichol phosphate mannose. And dolichol phosphate mannose is only part of the, if you go to your Essentials of Glycobiology textbook, every sugar has 2 entry points for going into macromolecules. GDP mannose is no different. It can come from glucose or mannose. And so here's what we've talked about so far, but what if we could have different ways of looking at this? And so we wondered about the possibility of glucose-derived mannose and mannose, exogenous mannose, salvage mannose, serving different reactions. And so this is a schematic of our interpretation of this. And as you can see up here, these molecules, these mannose molecules, are added on the cytoplasmic base of the ER. That molecule then is turned inside the ER where it picks up this. It picks up the dolichol phosphate mannose derivatives before it goes on to make that structure. But there's also a number of different kinds of proteins, such as GPI-anchored proteins, dystroglycan, and C-mannose proteins that only require the mannose that comes from perhaps a second pool. And you say, well, that's great. Do you have any evidence to back that up? And again, this is a very recent result. But in the absence of mannose, we can distinguish those reactions that require only GDP mannose over here. And if you look at which comes from glucose versus recycled mannose, that's salvage mannose. We did the same thing. We pre-labeled cells so we can see that difference. Now, if you look at GDP mannose plus mannose phosphate dolichol reactions, you can see that that ratio is very, very different than this one. And so our interpretation of this, it's going to require a lot more work and specific labeling, is that in fact, this pool is probably specific for, or at least enriched for being able to do those reactions in the interior of the ER. Now, to my way of thinking, this means that the cell is a lot more complicated than we think. Not only are there separate pools, but the Golgi that we see as those nice 4 or 5 stacks, it's not that simple. It's not that simple. It's sort of like looking at the intestine and saying, oh, it's an intestine. you have to go into the villi and the microvilli and, and all those things. I suspect that there's all this heterogeneity in the Golgi that we never knew about. And maybe we can't unless you have a technique like this where you can say, Golgi, do your thing. In Drosophila, for instance, there are separate Golgis. What if, in fact, when they merge into one Golgi, they still remember who they were? They have different functions. But we see them because we don't have high enough resolution. We see them as a single Golgi. Speculation, certainly at this point. And if that's true for mannose and for fucose, what about others? Galactose, N-acetylglucosamine, sialic acid. Are there separate pools that you might be able to use for O-gluconacylation, for instance? Different reactions. And I think we need to adjust our thinking. What's our conclusions? Well, I think you can probably read through those and think about them on your own. I won't bore you by reading because you can do that yourself. But again, I wonder if this is the tip of the iceberg. What are the heritage of other sugars? If we can measure these things in their products, because they're stable, we can separate that from just the flux analysis, which is typical of what you do in metabolomics. And what in the world is the basis of this selectivity? How does a cell know to do this? Where did it ever come from? So our future plans, we want to look at specific glycoprotein preferences. We're going to do this glycoproteomics differential labeling. What genes are controlling the degradation? Nobody's ever looked at that. And we're going to look at other ways to deliver fucose. You may know that you can grease up a sugar and have it go through the plasma membrane without having to run into a receptor, a transporter, or come through endocytosis. And those molecules tend to be a lot more efficient. So maybe they're bypassing some of the ways that the cell has designed to organize all of it. So with that, I'm going to stop and give an acknowledgement to the Rocket Fund for the funding, and I can tell you a story about that if you want to know it. NIH, of course, and Sanford Byrne Previs, where I've been for now 35 years. But this work was all pioneered by Paulina and ZJ, Bobby Yang, and Jamie as our lab assistant helping us out. And we have a whole bunch of collaborators that have made this work possible. So I'm going to stop there and take any questions if you have any energy left.